What do your gut and a mosquito's rectum have in common? The answer, neuropeptide Y receptors. Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. My guest today is Dr. Laura Duvall. She started her journey in a small town with no interstate highways and ended up uncovering how conserved neuropeptide-binding GPCRs regulate blood feeding, mating, and behavioral suppression in Aedes aegypti. You'll hear how a single GPCR mutation can override a mosquito's drive to bite, and how GPCR-driven behaviors in mosquitoes mirror conserved signaling we see in humans. Before we dive in, a quick word about resources that can deepen your GPCR expertise. GPCR drug discovery evolves faster than formal training can keep up. The Dr. GPCR University brings insider methods from leaders shaping the field. This is knowledge you won't find in textbooks. And if you've ever felt receptor pharmacology gets lost in translation between the bench and the boardroom, Theory's Corner is for you. Dr. Kanakin delivers real-world context and decision-focused lessons on what actually matters in drug development. This episode and the Dr. GPCR ecosystem are brought to you by our partners in GPCR research. Saltaris Research delivers innovative fluorescent ligand technology for non-radioactive GPCR assays that accelerate discovery workflows. Eurofins DiscoverX provides validated, industry-standard cell-based assays spanning discovery through regulatory submission, backed by 25 years of GPCR research expertise. All the links are in the show notes. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR, and I'm very excited to finally have Dr. Laura Duvall on the podcast. Laura, welcome.
Thank you so much. I'm happy to be here.
Very excited to have you on. I know we rescheduled this. I was sick and then things happened, so I'm glad we're finally doing this. All right, so let's start at the beginning. Would you please introduce yourself, tell us who you are and what do you do?
So I'm Laura Duvall, and I'm a biologist. I work in the Department of Biological Sciences at Columbia University, and I run a research lab where we're interested in understanding how mosquitoes regulate innate behaviors. And so we mostly look at mating behaviors and blood feeding behaviors in these animals.
Wonderful. Now, the next question is going to be what does that have to do with GPCRs? But before we go there, I'd be curious to hear how you got into science. Was it something from a young age, or did you stumble upon it?
Yeah, so for me, it was not something really from a young age. I grew up in a very rural part of the American South in North Carolina. It was a very small town. In fact, it was so small there were no interstates in the county that I grew up in. And it was the sort of place where if you were a smart kid, you were either going to be a doctor or a lawyer, end of list. And I was, pretty good at science and math. And in fact, I went away to a science and math magnet school in a different part of the state. And that for me really kind of opened up a lot of opportunities for me. But I still thought I would probably be a doctor because that was sort of the default option, career option for someone who had strengths in science and math. And it wasn't really until I landed in a work study job in a lab when I was in college that it really occurred to me that I could pursue research as a career. And this was mostly by chance, basically like working in a lab paid better than working at the gym or working in a library. And this was the first time that I really came into contact with people who were graduate students and postdocs. And what I also sort of realized at this time that was really important to me was how creative a career science could be. This idea that you could really kind of use a lot of creative elements in deciding which research questions to ask and how to ask those questions. And so that for me was really the experience. I stayed in this lab all 4 years when I was in undergraduate, and that was really the experience for me that made me want to apply to graduate school to pursue a PhD.
That is so wonderful. No interstate in the little town. Wow.
Yeah.
That's really cool. We will need to think about what we title this episode at the end. So I think that could be a very nice element that we can put into the title so that people get intrigued about what did we actually talk about.
Yeah.
So you worked in this lab during undergrad for 4 years, and then you decided to go to graduate school. I mean, research and science is vast. How did you decide which way to go?
So most of the time that I spent in graduate school, to be honest, was feeding brine shrimp to zebrafish. But I started out as a biochemistry major. And biochemistry is great, but as I was sort of going along in my coursework, I became more and more interested in neuroscience. And I remember reading this book. It's called Time, Love, and Memory. It's about behavioral genetics and Drosophila. And I got really excited about the idea of studying circadian rhythms because I thought there was this really elegant connection between genes and cells and behavior. And so by the time that I was applying to graduate school, I was applying specifically to neuroscience programs, and in many cases with a research interest in circadian rhythms.
Wonderful. I do remember my developmental, genetics courses, and I have to say that I was traumatized because I just thought it wasn't just. I was also in the biochemistry department, and I was like, oh,
Yeah.
I have to go to this class. But I have to say, I think the model system, the Drosophila model system, and the mosquito, it's just beautiful because it is, in a sense, simple yet complex enough, and you can manipulate the genes in a very elegant manner.
Exactly. And that was something that ultimately, when I was choosing a thesis lab, that was very appealing to me. So I ended up doing a thesis looking at how a certain neuropeptide called PDF, or pigment dispersing factor, regulates or coordinates clock cells in the brain of Drosophila. So this is also my entry to GPCRs. So go ahead.
How I understand that you picked the lab based on the topic because you were interested into that topic, but I'm assuming there's not only one lab that works on a topic you're interested in, and I think this is more of a question to help undergraduates or maybe graduates decide which way to go and how to pick a lab. What were other factors that you considered when you picked your PhD lab?
So there were a couple of things, and this is actually a piece of advice that I often give to people who are starting out in graduate school as well, which is that you have to figure out what scientific questions you're interested in, but also what experiments you get excited to do when you wake up in the morning. And so for me, I really loved having a lot of the genetic tools that we had in Drosophila. It made me feel like I really had a lot of access to be able to manipulate specific things, to really ask detailed questions. So I spent a lot of time in front of the microscope doing live imaging of Drosophila neurons during my PhD. And those were experiments that I just sort of loved to do. So it's not only this sort of, we often think of this as, what is the big burning scientific question that you have? But I think it's also really important to think about, what are you excited to get up and do in the morning?
Absolutely. And I'll add to that, who are you excited to do it with?
Absolutely.
Women counts as well.
Yeah.
Wonderful. So PhD, and then I'm assuming postdoc, because we talked about it before we hit record. You mentioned that this was, during the PhD was the time where you entered into the GPCRs actually entered your life. So maybe you can start by expanding on that.
Yeah. So GPCRs, my entry point was really because of their role in neurotransmission. And it was because we knew that there were these conserved neuropeptides that were important for regulating, in this case, circadian rhythms. And so what I was doing specifically in my PhD was sort of asking this question, which was, we knew that there's this neuropeptide. It's called pigment dispersing factor. It gets its name because it was first described for its role in regulating daily changes in pigmentation in crustaceans. So these shrimp will have changes in the pigmentation of their eye stalks. And so this is why we now call this pigment dispersing factor. But what we knew is that it's really important for coordinating behavioral circadian rhythms. And it plays this role in specialized clock cells in the Drosophila brain. Now, what was sort of mysterious is that these same cells co-express other closely related GPCRs. And in fact, those closely related GPCRs use some of the same downstream signaling components. Yet only one of them seemed to be able to coordinate the animal's circadian rhythms. And so one of my questions was really to understand how it is that the cell knows the difference between activation of this pigment dispersing factor receptor versus other closely related receptors in the same cells.
Wonderful. And this is where things get complicated because they have to share all the intracellular signaling proteins.
Exactly. And so what I was able to show is that there were different sort of signaling complexes that were assembled for these different receptors in the same cells.
Fascinating. So then after your PhD, you decided to do a postdoc. What motivated you to do the postdoc, and how did you pick your next, lab that you were going to work in?
Yeah. So at the end of my PhD, I'd had a good time. I was enjoying doing research, and I realized that I was really interested in pursuing these questions about how animals regulate these innate behaviors. And I thought that continuing to study neuropeptides would be a really nice way to connect that to behaviors in other species. So we know that these are often deeply conserved signaling pathways that you can find in animals, often ranging from, C. elegans to man. So I was interested in pursuing these types of questions. I also knew that I really liked working with invertebrates. And so those were sort of the types of labs that were on my list when I was thinking of places to do a postdoc.
That is really interesting. This is a completely side note. I was just watching a documentary and the question that they were trying to answer is why dogs are cute. Uh-huh. And it turns out they can move their eyebrows.
Yeah.
And that's what gives them these puppy eyes. And it made me wonder, obviously going back to GPCR, it made me wonder how did they evolve from the wolf to be that's not able to actually move their eyebrows, to the puppies and the doggies that can move their eyebrows, and what is that circuitry that gets activated in order to get to those puppy eyes, because they know what the human reaction will be to it.
There is this interesting, like, you can make like a baby face, right? Like you have big eyes and like expressive. And dogs certainly have like the wolf baby face.
Yes, exactly. So where did you go for your postdoc?
So I did my postdoc with Leslie Vosshall at the Rockefeller University. And to be honest, I showed up for this interview and I pitched her all of these ideas that I had about Drosophila research. And when I met with her, she said, well, I like you. These are good ideas, but my lab is becoming a mosquito lab. So if you want to come and do your postdoc with me, you can ask these types of questions, but you have to do it in mosquitoes. And at first, this was a little bit intimidating. But as I looked more into it, I realized that mosquitoes also have these very extreme sort of innate behaviors, right? All of us have firsthand experience with the fact that they're very attracted to find and bite humans. They also have sort of extreme mating behaviors where they basically have sex once, the females have sex once in their entire lives and then suppress their receptivity. And so I sort of came to appreciate that there were really interesting questions to ask in this species. And thanks to some of these genome editing tools like CRISPR-Cas, we really had an increasing arsenal of tools to actually ask those questions.
That is wonderful. And, as you know, I was also at Rockefeller University, and I've seen Leslie in action, and she's just phenomenal. She is such a powerhouse.
Yeah, no, it was a great place to do my postdoc.
And so what were you interested in? What were some of the questions that you were working on during your postdoc with this very new system?
Yeah, so here we were able to sort of continue the theme of GPCRs and narrow them in on a new family of neuropeptide receptors. In this case, we were looking at this neuropeptide Y family. And our interest in this came from the fact that this is a signaling pathway that plays really important roles in feeding behavior. And it turns out it also plays important roles in blood feeding behavior in the mosquitoes. And when I say that these are receptors that are conserved, one of the things that I mean by that is that we can actually take pharmacological compounds that were designed to target human receptors, and they will still crosstalk and activate the mosquito receptor. So this was a really important entry point for us because it meant that we could use these compounds and pharmacologically manipulate mosquito attraction to humans. And that told us that something in this family of receptors was important for regulating this.
That is wonderful. And people, peptide Y receptors are also, regulating or implicated in the regulation of feeding in humans as well. So it's directly translated into the human system.
Yeah.
A little bit more complicated, but I think having a nice mosquito system where it's quote unquote limited and you can actually do a lot of experiments, because if I remember correctly, there were a couple of people from Leslie's lab who I knew, and you guys were feeding mosquitoes just by putting your hands in there and just allowing them to just feed off of you.
Yeah, that still happens sometimes. But yeah, it turns out it's important to regulate when you want to feed and when you don't want to feed, if you're a worm or a fly or a mouse or a human, and that we're still using some of the same pathways to do that.
Wonderful. And then during your time in the lab, and what were some of the, major highlights? What were some of the major discoveries that you made that you felt like were the motivation for you to get into an academic position and open your own lab?
So one of the major discoveries was that we identified the identity of the mosquito receptor that was important for regulating. Now we call this host-seeking suppression. What I should kind of provide as background here is the fact that everybody knows that mosquitoes love to find you and bite you. What you may not know is that they don't want to do that all the time. And in fact, the female's internal state can really change her desire to bite humans. So after she's taken this big meal of blood, she'll actually use that protein from your blood in order to develop eggs. And so she'll spend 3 to 4 days digesting that protein, developing her eggs, and largely suppressing her attraction to bite humans. And so it turns out that this receptor that I characterized in graduate school is really important for that. And so one really exciting day for me was when I went in, used CRISPR-Cas tools to actually mutate this receptor and show that these females continued to inappropriately host-seek even after they had a blood meal. Right, this feeling of making, this mutant mosquito disrupting a single gene and changing this incredibly strong behavioral drive was something that was, almost magical, right?
Absolutely. And now I don't have any experience working with mosquitoes at all. I'm the cell culture Hexcel type of, scientist. But what did that look like? Like, what are the steps more in details that you took? I really want to get a sense of what does that experiment look like? You go into the lab and you say, I'm going to CRISPR out this gene, and what do you do? I think the audience might enjoy hearing more about that.
Yeah. So one of the first things that we need to do is actually really make sure that we have a handle on what the gene is. For a long time, the mosquito genome, it's sort of an absolute nightmare. It's the size of basically the mouse genome. It's full of highly repetitive elements. So we have to make sure that we know exactly. Oh, wait, sorry. Give me 1 second.
Oh, take your time. We can edit this out.
Sorry, I had an alarm going off in a bag.
No worries.
I
I couldn't hear it.
Okay, good.
No, not at all.
All right. So, what this looks like for us when we want to CRISPR a gene in mosquitoes is that we find our gene of interest. We make sure that we know exactly what we want to target, and then we design guides that can cut this gene. And at this point, now in the old days, this was like 2013, 2014, we were really excited if we could just disrupt the gene, if we could just insert or delete something to mess up the gene. Nowadays, we sort of usually do higher tech things where we're either knocking in some kind of a fluorescent reporter or something that lets us turn this into a driver line.
Wow.
And so then we get these mutant mosquitoes. At this point, we usually have some kind of a fluorescent marker. This means that we have these sort of adorable mosquito larvae that have bright green eyes. So we can make sure that it's expressing our transgene of interest. And then what we do, now the old school way of doing this is when I was a postdoc, is that you put them in this assay that's called the uniport olfactometer, which is a very fancy name for something that is a very simple assay. It's just a tube with hungry mosquitoes at one end and my arm at the other end. And we let the mosquitoes go, we give them 5 minutes, and then we collect them in a trap that's right next to my arm. And so it's very simple, but it's incredibly sort of exciting when you can make a manipulation that actually turns off this drive to find and bite humans.
Yeah, you can sense it. You have the results then and there, and you can sense it on your own arm.
Yeah. Now, I mean, one of the things that I'll say is that we've now been using, like a lot of people who are studying various aspects of behavior, we've now really more commonly been moving to a lot of video tracking assays and using some of these machine learning algorithms so that we can get sort of a more rich data set. But there's nothing more satisfying than just sort of sticking your arm in a tube and seeing if you can get them to turn down their attraction, to turn off their attraction to your arm.
That is lovely. I want to hear a little bit more about the machine learning and the video recording of the mosquito behavior. Because when I think about it, they're tiny. And you need to be able to monitor them. And I'm assuming you don't want them to fly around in the lab. So they have to be in specific compartments, and you'd have different mutants. I'm assuming, what does that look like? What are some of the questions you're able to answer now with a video recording and the machine learning algorithms that you were not able to do previously?
Yeah, so this is something that, let me think. We've been using these for a number of different assays. And one thing that I should mention is that, mosquitoes are still a little bit the Wild West, which means that most of the time, if we want a new behavioral assay, we are designing it ourselves and we are building it ourselves. And one of the new assays that we've built with this has led us back to some of our interests in circadian rhythms, and it lets us ask questions about host-seeking behavior at different times of day. So this means that we can load a group of mosquitoes, they can be wild type or they can be mutant, and we can apply host stimuli using an automated system basically throughout the 24-hour day. So the fact that this is automated means that when we want to ask if they're interested in biting us at 4 o'clock in the morning, we can just program that into the code, and we don't have to have, the graduate students and the postdocs don't actually have to be in the lab at 4 o'clock in the morning. So this lets us look at responses in a way where the human is not even in the room. That's something that's important because in a lot of our experiments, not only are you the experimenter carrying out the research, but you also might be an accidental stimulus. So by allowing us to deliver these host-associated cues in an automated way, it lets us ask these questions in a way where the experimenter is not in the room, and it lets us ask this, like I said, throughout the 24-hour day and under environmental conditions where, for example, the lights are off. You wouldn't want to sit in a completely dark room at 4 o'clock in the morning with your arm in a tube.
Definitely not. And as you know, I'm sure you know, and a lot of people know this, mosquitoes sometimes tend to prefer one human versus another.
Yeah.
And so that can also, maybe you'd have the graduate student that never gets bitten by mosquitoes, that don't like them, that would also skew and really bias the experiment.
No, this is true. And we sort of have to keep this standardized because we do know, and this is work from Ellen Deobaldia, who was also a postdoc with me in the Vauxhall lab. Now, everyone has this anecdotal experience that, I go hiking and my mother always gets bitten, but I never get bitten. And you also have this experience that's relatively stable over time. It doesn't depend on what shampoo you used or what soap you used. And we know that this is true. People do have different sort of levels of baseline attractiveness to mosquitoes. And so we do have to control who is the stimulus in our assay. Most of the time it makes sense to use yourself as the stimulus. I am one of these people who's incredibly unattractive to mosquitoes, which sometimes makes my experiments more complicated, but is good in real life.
Yeah. Wow. Wonderful. And so going back a little bit to the timeline, you're almost done with your postdoc and you're thinking about next steps. This is also thinking about the listeners, PhD students, postdocs, maybe thinking about their next career jump. How did you decide to go to look for an academic position?
Yeah. So for me, this idea, now I will say that I am very excited about just the basic biology of mosquitoes. And so the idea of looking for an academic position where we could continue to pursue these questions was really exciting to me. The other thing that I was thinking about is that we were starting to find that we had some questions, or I was starting to find that I had some questions that were not necessarily strictly neuroscience related. My PhD was in neuroscience. That had been a lot of the focus of my previous work, but I'm in a biology department now. And that's something that I really find a lot of value in, because it means that there are different directions that we can pursue, even if they're not kind of strictly neuroscience questions.
I love it. I really love it, because actually my next question was going to be, whenever you design a project or you think about a new project, how do you approach the translatability, I don't know if that's even a word, to other systems, mainly the end goal being human systems? And what you learn in the mosquito, how can you use that information in order to learn more about the human system?
So there are sort of two facets to this. One facet is to think about how the biology that we uncover in the mosquitoes might be related to biology that's also happening in humans. And I'll tell you a really interesting example of something that we have recently discovered where there were some surprising parallels between mosquitoes and humans. And so this was in following up on this neuropeptide Y related receptor that I characterized as a postdoc. One of the first projects that we really wanted to start in my independent position was to understand more about how this receptor was working, where it was expressed, and already this has taken us into a really surprising direction. So our first question was just to look and see where we find the transcript for this receptor. Where is it expressed in different parts of the mosquito anatomy? And it was really hard for us to find, because we sort of thought that it would be expressed in the brain. It's a neuropeptide receptor. But in fact, it seems to be expressed exclusively in the mosquito rectum.
Wow.
Yeah, a tissue that I did not know existed until we got this result.
Wow.
And so one of the things that we've been doing now, and so at first we thought this was like a really surprising result. Like, what is this neuropeptide receptor doing in this weird part of the mosquito gut? But it turns out that you and I have neuropeptide Y2 receptors in our colons as well. And it's also really important for things like gut motility. And so we've been starting to sort of follow up more on thinking about how these cells might be receiving signals and how they might be sending signals to the rest of the animal. And we've done some work that suggests there are transcripts associated with vesicle release and neurotransmitter synthesis in these epithelial cells in the mosquito rectum, which sounds totally bizarre. And yet you and I also have non-neuronal excitable cells in our guts that are capable of making and releasing neurotransmitters. And so really, go ahead.
Sorry, no, please. I think it's wonderful. And it a little bit reminds me of the bitter taste receptors. When you hear taste, you think mouth, tongue.
Yeah.
But these guys are expressed in the gut as well and in other parts of the body.
Yeah.
So it's really fascinating. We know a lot, but we don't know anything.
Yeah. And these things are, they're not necessarily doing the most obvious thing. But really, we've been finding a lot of inspiration from, papers on neuropod cells or enterochromaffin cells in mammals that have been really guiding some of the next experiments that we're going to do. So that's sort of one example where we've had kind of a basic biology finding that has really kind of interesting parallels with what you see in mammals and in humans. Now, the other aspect of this, to think about how it relates to humans, is basically to think about the potential to disrupt these behaviors in mosquitoes and use that as a way to prevent pathogen transmission. So one of the things that you can imagine, I should mention that the species of mosquito that we mostly study is called Aedes aegypti. And so this is a species that is usually called the yellow fever mosquito in textbooks, but really she makes the news these days for her ability to transmit pathogens that can cause things like Zika and Dengue and chikungunya. And one thing that you can imagine is that if we could find ways to sort of weaponize these pathways that are naturally suppressing mosquito attraction to humans, to turn them on and to turn these pathways on and to turn off attraction to humans, that's something that could potentially disrupt the transmission of all of those pathogens. So instead of having a Zika specific therapeutic or dengue specific therapeutic, if you could disrupt this behavior, it would sort of stop things at the point of transmission of all of them.
Wonderful. Quick question. How does the attraction to humans, of mosquitoes to humans, compare to their attraction to animals? I'm thinking dogs here.
Yeah, this is something that varies a lot by mosquito species. There are thousands of mosquito species. Only a few of them are the ones that you really read about as major contributors to disease. And that's because there are some of these species that have a strong preference for humans. And this includes the Aedes aegypti mosquitoes that we study. So what this means, and other people have shown this, is if you give these animals a choice, if you give these mosquitoes a choice between biting, let's say, a human or a dog, they'll strongly prefer the human. But that's not true. There are some species that are much more sort of generalists and would go sort of equally to either animal, which means that those animals, in many cases, or those mosquitoes, in many cases, are much less effective at spreading pathogens among humans.
Interesting. That is very interesting, because I've been wondering, you go out on a walk with a dog, and then there's, when you're in the woods or close to the woods and there's mosquitoes, and then you spray yourself with bug spray, but what about the dog?
Yeah. And we know there are some species that actually prefer to bite birds. So the Culex mosquitoes, that can transmit West Nile, if they would usually choose a bird over a human. But for the most part, if you're around and you're, easy to bite, most of them won't turn down a human.
Lovely. I love it. Is there any, from a, let's say, neuronal or GPCR receptor perspective, or what is it that drive these mosquitoes to have a preference for a human over any other animal?
So
is it known at all?
So this is probably something that's more related to their peripheral chemosensation. So in fact, Lindy McBride's lab at Princeton has been really interested in understanding this evolution of human preference. And so we know that different species of mosquitoes are expressing sort of different complements and different levels of different odorant receptors that allow them to be tuned to detect human odor. It turns out that humans and animals, if you could sort of look at the chemical composition of what we smell like, are actually using a lot of the same chemicals, but the ratios and the levels of these things can be quite different.
And so the mosquitoes can make these subtle differences between, wow.
And even between humans. This is sort of where some of the basis of human preference also comes from. We know that it's the way that you smell that sort of determines how attractive you are to mosquitoes.
That is so interesting. Kind of as a side note, and going back to the smell, I have a friend who was telling me that he was going to hunt with someone who's, used to going to hunting, and he was told, do not use any deodorant, do not use anything, and here is the soap that you should be washing yourself with for the next 2 weeks before we go hunting, because if you use anything other than this, the animals are going to smell you.
Yeah. I mean, they're explicitly attuned, right, to all of these cues that we may not be paying so much attention to.
I thought it was really fascinating, the sense of smell, and these are the things that we don't typically think about.
Yeah.
Right. A lot of interesting things to study. Now that you have established your lab, and I have a very naive question. I mean, I never think about, okay, if I want to work on mosquitoes, where do I actually get the mosquito species in order to get what does that require in order to start a lab and work on mosquitoes or flies or what does that look like from an equipment perspective? Just maybe give the audience an idea of what did your day-to-day look like in the beginning when you started your lab?
Yeah, well, I will say that I started my lab in 2020, so right in the middle of the pandemic. So it looked very chaotic, is the short answer. But I can tell you that we use what is essentially like the lab mouse version of mosquitoes. So we have inbred laboratory strains that we use. We use these when we make a lot of our transgenic animals because we have a very good idea of what their genome is like. We do not have any pathogens in the lab. But we still have to think a lot about mosquito containment, right? We would be very unpopular, certainly with all of our neighbors, if we were not good at mosquito containment. So there are some practical considerations. This means that between the general lab space and the mosquitoes, we always have to have 2 barriers. So you sort of go through what looks like an airlock when you come into the mosquito facility. And because we're mostly working with a tropical species of mosquitoes, we house them in what looks like a cold room. But when you step inside, it actually feels like the worst day in August. So we like to keep this at 28 degrees Celsius and 80% humidity. So that's where we have our, tens of thousands of mosquitoes, all contained in mesh cages. So they're not, you're not immediately attacked by mosquitoes as soon as you walk in. But that's how we mostly keep them and house them. It's sort of DIY. So mosquito larvae are actually aquatic. You probably have this experience that mosquitoes lay their eggs in things like abandoned flower pots or bird baths in the yard. And the early developmental stages are actually aquatic. We rear these actually in restaurant pans. So we have these restaurant pans that we sort of mimic stagnant pond water by giving them some fish food. And then about a week later, we can use a tea strainer to collect them in cups and then let them become adults inside the cage. So that's what day-to-day life looks like.
I love it. This is the first time I get to ask this question, and this is the first time I actually think about asking this question. So thank you so much for giving me a visual walkthrough and what that looks like on a day-to-day basis.
Yeah, I will say we also have just gotten in some field-collected mosquitoes. So this has been sort of an adventure. I laugh because I knew that this was going to be the worst field work in the world because we were sending people down from the lab to go and basically stand in ditches in Florida in the middle of the summer. But people were very excited to do it. They felt like it was very glamorous to having like a field work element. And so those mosquitoes, we've also brought back into the laboratory. So we rear them in the laboratory. We make sure that they're not carrying any pathogens. But that's something that's sort of interesting to us because it will allow us to test to see whether or not the discoveries that we make in our inbred lab strain, where we can make the transgenics, also hold up in real mosquitoes that we've collected from the field.
Super interesting. Wonderful. Let's get to inspiring junior scientists a little bit.
Yes.
Top 3 aha moments that you had as a scientist that shaped your trajectory.
One for me was definitely feeling like I had made a finding in graduate school, realizing that I had collected a complete set of data. And in this case, this was looking at these responses of these clock cells and realizing that, like, from the ground up, I had made a finding that no one in the world had ever discovered before. That was a real aha moment for me. I would say another one was when I was a postdoc and really seeing behavioral phenotypes in the mosquito mutants that I had made. It was just incredibly satisfying to see that manipulating a single gene was able to really change such a robust behavior. And then I would say another sort of aha moment for me that's not exactly related to being in the laboratory was going to some of these conferences when I was a postdoc, some of these neuroethology conferences, where I remember having this moment, listening to some of these, absolutely fascinating talks on, navigation in bats and mating behaviors of these different insects, and realizing that this was my job and that this is a community that I was a part of. And that was a really exciting moment for me. And for me, that was a moment of really feeling like I wanted to run my own lab.
Wonderful. And now you're part of another community, which is the GPCR community.
So absolutely. Yeah.
We're a friendly bunch. We are.
Yeah.
Thank you for that. That's very interesting. Especially, I think, the aspect where you mentioned that you realized that you had a question, you collected all the data, and you were able to go through that entire process, and you were able to synthesize your findings and say, wow, I just found this.
No, exactly. I feel like you kind of go through this progression, right? Where first you realize you can, do an experiment and get some data, and then eventually you get enough data and things, and you sort of realize that you've made, a new finding.
Yeah.
And that to me was sort of getting to that level of, beyond just I'm collecting the data to realizing sort of the bigger implications of those results.
I think so. I think it's wonderful. And I think another one in general, not necessarily for you, but I think I hope it will resonate, is when you're in the lab and you're realizing that you're asking questions that are your questions.
Yes. Yes.
It's when you're saying, wow, I think now is the time for me to move on, because now I have gotten to a point where I can run the experiments, I can ask questions, I can find things, but now I want to ask larger questions that don't necessarily cover or don't necessarily fit anymore in my postdoc lab or in my PhD lab.
And I think this also gets to something that I had mentioned earlier about realizing what a creative profession research can be.
Absolutely. Absolutely. And I think it comes also with experience.
Yeah.
And with confidence. Experience builds confidence, and then that confidence gets you out of that comfort zone.
Yeah. Yeah. And I think the other thing that I will say that I find really exciting about the type of research that we're doing is that it puts us at the interface of a couple of different fields. And I just find, whenever I present our work to different audiences, I get so many new ideas because there's so many things that I've never thought of that people really come up with, because of their different perspectives. And so I really appreciate being at the intersection of a number of different fields.
And you make a great point, because I think, in your case, working with a model system that's mosquitoes, asking very specific questions and not being on, I want to say mainstream, hex cells and breath assays and those response curves and doing the classical in vitro pharmacology assays, it can be really nicely complementing your research as much as, the in vitro pharmacologist running experiments in a hex cell can benefit from looking at potentially their GPCR's activity in a model system.
Yeah. No, that's something, even when I was in graduate school, this idea of sort of bridging this idea of looking at genes, looking at cells, looking at behavior, seeing if we can actually make manipulations and connect all of these things is something that I find really satisfying.
I agree. I have a little visitor. If you'll excuse me 1 second.
No worries.
I will be right back.
Yeah.
Apologies.
No worries at all.
It wasn't a timer. It was a different kind of timer.
Yeah.
Wonderful. Any advice to junior scientists who might want to contribute to the field?
One thing that I wish I had learned a little bit earlier when I was in graduate school was that you really can drive your own research interests. I think I underestimated my own expertise. I thought that I had this very narrow set of expertise. And it really, when I moved into mosquitoes for my postdoc, I realized that an important part of your training is realizing that you can generalize and you can build expertise in a new field. And I think that that's something that can make for some of the most interesting scientific careers.
I agree. And I think it also, again, it comes with experience, that confidence.
And related to that, I would say get into a lab and ideally experience more than one lab. So my day-to-day life is quite different from many other people who are biologists, many other people who are neuroscientists. Some people may spend all day coding in front of a computer, others may be in front of the bench, other people may be out in the field. And really getting into a lab and ideally getting into, having experience in more than one lab, I think is something that's really valuable to help you to realize what your research interests are.
I agree. And unfortunately, in my case at the time, and I think this is in the US system, you get to do rotations and you get to work in different labs. And I think that's very important. At the time at the University of Montreal, you wanted to go to graduate school, you applied for a master's, you picked your lab, and that's it.
Yeah.
And it was very difficult to say, after 3 or 6 months or any amount of time to say, no, I need to switch.
Mm-hmm.
So it was a hit or miss for a lot of people. And I know a lot of people who just left research and science in general because they had that bad experience.
Yeah.
It wasn't a good fit. And that happens. And that's okay.
Yeah.
Not being able to make that transition can be very devastating for the field.
Yeah.
You're right. I think that's important. And I think that the fact that you get to work in different labs allows you to work with different investigators, different team members.
Yes.
That can influence you positively.
And the other thing, and the other thing that I think is really important is that often people think about what is the PI like if I'm going to join this lab? And that's important, but in some cases, your sort of intermediate colleagues, so people who are other graduate students, people who are postdocs in those labs, are incredibly valuable resources. For me, as someone who's gone, throughout my training, the people that I still talk to people who were postdocs in my graduate lab, colleagues of mine. So the PI is very important, but also I would say don't underestimate the value of your colleagues as well.
I agree. And I tell people it's when you go into interview in any situations, whatever job you're looking at, you are interviewing them just as much as they're interviewing you. And it's kind of like dating in a sense, because you're going to spend a lot of time together.
Yes.
And if you feel like you can't learn from those people because for XYZ reason, or you don't feel comfortable, or they don't feel comfortable with you, then that hint or that gut feeling has to be taken into considerations. In an ideal world, you want to be in an environment where everybody is helpful, where you can help mentor the newest person, and when you are also mentored, and not only by the PI.
Yeah, and I think you also sort of want to feel inspired to to really reach the next stage of your career. Even being a senior graduate student is very different than being a graduate student who's just starting out.
Absolutely.
And seeing that development, I think, is also, you want to see people developing in the lab.
Absolutely. You want to be able to see people say, hey, wait a minute, I can show you because I was shown a year ago and now I know how to do it.
Exactly.
Let me show you. Let me teach you. Absolutely. All right. Two last questions. What is your favorite GPCR? Because I forgot to ask it before.
I think my favorite family right now is the Neuropeptide Y family because it's both so conserved across so many organisms and because it seems to be so uniquely specialized. So in this case, this is a receptor that's expressed in the mosquito rectum.
I think it's so interesting that it is expressed there. And it makes me wonder, even in the human gut, what does it do? Why is it there? And how does it relate with the other members of the family?
Well, I think it's also, made me think a lot about the fact that in some ways, my view of neuroscience and signaling between the brain and the body is relatively narrow. It's really important to have these sensors everywhere in the system. And so I think that this is something that has probably been underappreciated.
I wonder if I agree with you. I think it might have been underappreciated, but I wonder if it's because of the tools or the lack of tools, or we were not able to make those observations. Therefore, we were not able to make those connections.
Mm-hmm.
Where there's that extra little piece. Before we hit record, we were talking about the Gordon conference you were at, and I was talking about the endosomal signaling.
Yes.
And it's important. It's always been there. We were never able to see it up until a couple of years ago. And now we're happy that we can see it. But the question is, and so what?
Yeah,
what does it do to the function of the receptor? And how could we potentially leverage that for drug discovery purposes?
Yeah. And one of our big questions is, how is this receptor acting in this sort of unexpected cell type? Is it behaving in the way that we think that, a GPCR should sort of canonically behave? Or is it doing something slightly different? And so for us, I think there's some more tools to build. But I think that that's really going to be an interesting next set of questions for us to pursue.
I love it. It also reminds me of adhesion GPCRs, which is also this huge question. You can have 600 to 6,000 amino acids, and then you have this very long N-terminus that has all these domains. And the question becomes, okay, so what's the goal of having all these?
So why? Yeah.
Yeah. Is it mechanosensing, or does it get activated like your classical beta-2 adrenergic receptor or rhodopsin, or what does it do? Although rhodopsin is a whole different category now that I, said it out loud.
But yeah, there's no shortage of questions, right? We will have GPCRs. I haven't even gotten into all of the mating stuff. We will have behaviors regulated by GPCRs to study for my entire career.
Wonderful. I love it. I can't wait to hear about it. All right, last question. I think this is the toughest one. What should we title this episode?
Oh,
I know. At a high level, I think maybe if I can quickly summarize what I internalized from the conversation that'll help us maybe get to that point is that obviously GPCRs are everywhere.
Yeah.
And you can study them in different model organisms. And in the mosquitoes, it does regulate feeding behavior and mating behavior, which we didn't get a deep dive into it. But what I find fascinating is the fact that exactly this NPY receptor that you were mentioning, it's localized expression, and that it also translates to it has its own, it has its, let's say, its cousin in the human gut.
Yeah.
I wonder what we should be titling this episode. Depending on the guest, it can be anything. It can be very specific to the science. It can be also specific to the person. So,
I mean, I will tell you that when I give and we didn't get to the lust part of this, but I often introduce my general talks as lust and gluttony and mosquitoes. Ooh. We just got to the gluttony part. But
let's talk about lust a little bit if you have a little couple of more minutes, and then we can include that, and maybe we can give it something more, a spin to it.
Okay, so the motivation for this is that there is this book, a textbook on neuropeptides, which has the greatest opening line of all time, which is, it is thought that at least 3 of the 7 deadly sins are mediated by neuropeptides.
Most likely GPCRs. There you go.
Exactly. And so I like to think that this is an entry point for us. My goal is to get to maybe 3 of these by the time I go up for tenure. The mating part of this is that it turns out that one of these NPY-like receptors seems to have been repurposed as a receptor in the female that detects that she has mated with a male and seems to be important for suppressing her receptivity to mate with other males. Mosquitoes, sort of like their blood feeding, are also extreme in the way that they mate. So females mate once in their entire lifetime. And by mate, I mean have sex once in their entire lifetime. It's not like it's a monogamous pair of male and female. And we know that the males are transferring signals to the females that include neuropeptides that can activate targets in the female to change her behavior. And we don't know all of these. There's still a big mystery as to what the signal is that actually changes the female's behavior for the rest of her life. But we've identified one of these signals that's important for sort of short-term regulation of her mating. And it turns out it's a peptide from males that activates one of these GPCRs in the female.
Wow.
And so this is interesting because if you can mutate this receptor, then what you end up with are females who can mate with multiple males. And if you could exogenously activate this receptor, you could basically turn off female mating behavior, which would be another sort of indirect way to prevent mosquitoes from reproducing.
Wow. It is fascinating. And I don't know which part am I fascinated most about. It's a once in a lifetime, or the fact that the neuropeptide from the male activates the receptor in the female. Wow. It is wonderful. To be honest, I never spent enough time thinking about mosquitoes other than adding bug spray on me and the children.
Yeah.
But I think it's just wonderful. All of these questions, there's more questions that need to be answered for sure.
Yeah, and we still have lots of questions about, like I said, what's mediating the long-term effect, what's changing. We don't even know what all the signals are from the males. We don't know how all those signals are detected in the female, and we don't know how the neural circuits might be manipulated in the females. So all of these are questions that we're interested in.
Wow. All right, let's get back to the title.
Okay.
Knowing this. Well, I definitely like the lust and gluttony aspect to put into the title, but good.
I mean, there's also sort of like a gut brain, like that's another connection that.
Yeah, with the NPY expressed in the. I am lacking inspiration today. After a week of vacation, I have a hard time getting back into it. I think let's call it the gut, the brain, and we need something that ends like the good, the bad, and the ugly.
Yeah, or like
the brain.
GPCR regular. I mean, it's like I think we need mosquitoes.
Yes.
GPCRs.
Yes. GPCR, mosquitoes. So I have GPCRs, mosquitoes, and lust.
Let's see. Blood. I think of like.
People would be like,
blood, sex, and GPCRs, right? Like,
ooh, I love it. But yeah, blood, lust, and GPCRs.
How GPCRs control, I don't know, mosquito lust for your blood, appetite for your blood, mating.
Lust for your blood. I like that. What do you think? It's I think we need a subtitle for this because lust for your blood might sound very
I don't know if this is gonna go on a platform where it's gonna be like flagged for explicit content.
Yeah, I know. Okay, maybe we should tone it down, but still it's fun to play around with these words.
Yeah.
So, mosquitoes, GPCRs, and gluttony. It's no.
Or the drive to. Sometimes I say that they hunt humans.
Yeah. How or why mosquitoes? How GPCRs direct mosquito hunt humans.
How GPCRs
drive mosquitoes
drive to bite you or to
mosquitoes, yes, to humans. How GPCRs drive mosquitoes to humans.
Yeah.
Simple. Although I did like the lust for blood, but I think if we put this on LinkedIn and Twitter, it might get flagged for explicit content.
Yeah, this was I had to give my practice tenure talk, and I did the whole intro with the 7 deadly sins, and then I was like, these are the 2 that we're going to go out and talk about today. And for that audience, which was pretty informal, it killed, but probably not for the title.
No, definitely not. So how GPCRs drive mosquitoes to humans.
Yeah.
Simple, scientific, serene, and then everybody who's going to listen and watch this episode is going to hear all of the variations that we got through.
Yeah.
to find this title.
Okay.
I'll stop recording and then we can continue our chat. But Laura, it was really a great pleasure chatting with you. I really appreciate your time, and I'm so happy we got to see each other.
Yeah, it was great to see you, and I love talking about this stuff. So thank you so much for having me.
Very, very, you're very welcome. I'm happy to have you on board. Don't go anywhere. I'm going to let people wonder what we're talking about after this.
That was Dr. Laura Duvall from Columbia University. Her work reminds us that GPCRs are not just receptors in abstract. They are behavioral switches that orchestrate life or death decisions. From host-seeking suppression to lifetime mating choices, Dr. Duvall's work shows us how conserved signaling pathways shape survival strategies across species. If this conversation expanded how you think about GPCRs, here's how you can help others discover it too. Share this episode with a colleague, subscribe to the Dr. GPCR podcast, and leave us a 5-star review. It helps scientists like you find this community. Do you want to keep learning? The Dr. GPCR University brings you insider methods from leaders shaping the field. Subscribe to stay ahead of the curve. And Terry's Corner delivers real-world context and decisions focus from Dr. Terry Kanakin on what actually matters in drug development. This episode was made possible by our partners in GPCR research. Thank you to Celtaris Research for advancing fluorescent ligand technologies that are accelerating discovery workflows. And thank you to Eurofins DiscoverX. Your validated platforms and 25 years of expertise are advancing GPCR-targeted therapeutics from discovery to regulatory submission. And of course, thank you to the Dr. GPCR production team for making this conversation possible. Because receptors don't work alone, and neither should you. Until next time, stay safe.